This animation is designed to help you understand various ways that EEG and ERP data can be illustrated in research papers. Often figures showing EEG and ERP data can be confusing because there are a number of different ways that researchers display their results. The purpose of this video is to show you the connection between the neural signals generated in the brain and the graphs that researchers use to convey the properties, timing, and locations of those signals. We begin this animation by showing you how the neural signal can be visualized using event-related potentials, or ERPs. Here the neural signal is represented as dipoles in the brain. The positive side of the dipole is indicated in red, and the negative side of the dipole is indicated in blue. However, when we use ERPs, we aren't looking directly at this activity in the brain. Instead, we are looking at the electrical activity at the scalp, generated by those dipoles. We can visualize this electrical activity in three ways. First, we can visualize the brain's electrical activity measured at a single electrode. If we look at the brain shown here, we can represent the neural activity at the electrode shown here by plotting the amplitude of electricity in microvolts over time in milliseconds. In this particular graph, the red positive end of the dipole creates a positive amplitude, and the blue negative end of the dipole creates a negative amplitude. Here, positive is up and negative is down, although sometimes researchers plot negative amplitudes up and positive amplitudes down. This graph shows the level of electricity at the scalp starting at the time negative 200 milliseconds. The y-axis crosses the x-axis at 0 milliseconds, the moment when a stimulus is presented. Thus, in this graph, changes in the electrical signal are plotted over time from negative 200 milliseconds to positive 800 milliseconds, or just under 1 second. The y-axis is the positive or negative electrical signal in microvolts, also known as the amplitude. In these images, we are looking for specific deflections in the waveform, also known as components, when they are associated with specific cognitive processes. For example, in this image, the peak or deflection occurring around 300 milliseconds, known as the P300, can be seen. Thus, this type of waveform graph shows electrical activity over time at a single electrode. ERP data can also be depicted as electrical activity over time at multiple electrodes. For most EEG or ERP experiments, we record simultaneously from multiple electrodes positioned across the scalp. To accurately represent differences in electrical activity at different locations on the scalp, we need to plot each individual electrode as can be seen here. Each electrode has a name that represents its location on the head. You can see that the components are more visible in the electrical activity of some electrodes as compared to others. For example, the P300 can best be observed from the PZ electrode, where PZ stands for Central Parietal Electrode. Thus, plotting ERP data this way allows us to see neural activity over time for all electrodes. Nonetheless, you can also see that plotting neural activity across the brain in this manner makes it difficult to mentally combine all the different plots of activity to understand the topography of the activity. To help make it easier to understand the distribution of electrical activity across the scalp, researchers also plot ERP data in topographical maps. One way to see the distribution of electrical activity across all the electrodes is to present data at a single point in time, for example 400 milliseconds, and color code the amplitudes at that time across all the electrodes. Red indicates positive deflections, blue indicates negative deflections, and color intensity indicates the size of the amplitude. In this image, instead of a graph, we see a head plot that shows the location of the electrodes on the scalp. However, instead of showing electrical activity over time for one electrode, this head plot shows a map of activity at a singular moment in time across all of the electrodes. Thus, this image represents activity at 400 milliseconds, a single point in time, and at that time there are positive amplitudes at the back of the scalp over the occipital lobe. Note that there are also negative amplitudes that are located towards the front of the scalp over the frontal electrodes. Thus, this graph depicts the electrical activity across all the electrodes at one point in time. Nonetheless, we can also use this type of graph to show the distribution of electrical activity across the scalp across multiple time points. In this graph, we have selected time points every 50 milliseconds. You can see that positive activity in the occipital regions develops between 250 and 600 milliseconds. Note, in this type of graph, we lose the time resolution of the waveform graphs, but gain a better sense of the topography of the activity over time. Thus, the same ERP data can be represented in two ways as seen here. Actually, there are several other ways to visualize this information, but they are typically variations of these representations or different ways of representing the same information. Waveform graphs show you detailed information about changes in electrical activity across time as measured by a single electrode. Groups of waveform graphs show you about electrical activity across multiple electrodes to give you a better sense of the distribution of electrical activity across the scalp, but it is still difficult to see the topography of the activity. A topographical map shows you the distribution of electrical activity across the scalp at a single time point, but groups of topographical maps show you changes in this distribution over segments of time. Which type researchers use depends on the most important aspects of their data they wish to convey.